All right, everyone, welcome back for the afternoon segment of our symposium. So we'll begin with more year one awardee updates. And just for our year one awardees that are going to be presenting in this section, as just a reminder, you'll have 20 minutes for presentation, 5 minutes for questions. I will be sitting in the front and give you a 5 and 1 minute warning with my hands. And we'll go ahead and get started with Dr. Ramkumar Mathur from the University of North Dakota.
Hello, hello everyone. My name is Ram. I'm from Department of Geriatrics and University of North Dakota. So we do immune aging, like what is the molecular mechanism of how the immune system get like decline with the age, and it associated with colon cancer or neurodegenerative diseases. So here I'm going to show today how this immune system, like how the tumor microenvironment actually drive the immune system and dysfunction, like how this even tumor microenvironment like affect the immune system and suppress it in the colon cancer condition. So we know like this is the colon cancer is a start from the epithelial cells. You see and it's like more progressive disease is stage 1, stage 2, stage 3. Stage 3 just goes to the tumor cells go to lymph node, and then stage 4 is actually like metastasize, and then it get like more lethal. And as you can see, colon cancer is the second largest leading cause of the cancer-related disease, and on average 1 to 20 of people are risk. And as you can see, like 90% of new cases occur in the 50-year-old or more, like people. And as Gary mentioned this morning, it's more concerning is like in North Dakota, as if you can see, compared to national average of North Dakota has pretty high risk of getting colon cancer. So definitely it's like a concern for North Dakota, to look at the what is the colon cancer and like how you can treat better. The problem is you can see this colon cancer is like very complex, and the intestine is like already very complex. It's like a microbiota, and you can see like over here uh, how this tumor microenvironment is like epithelial cells, infiltrating monocyte, inflammatory cells, T cells, and this immune environment is like very complex. And most of the study, like traditional study, has shown in the 2D cell culture or in vitro cell culture, or showing some how the epithelial immune axis and what there is like the significant gap, looking at the uh, cellular level or like single cell resolution level, how the epithelial or epithelial subsets are interacting with the T cells or CD8 T cells, how they're defining the uh, epithelial immune system is like really is not very much shown. And there are a lot of mechanisms show like how the post-translation modifications such as phosphorylation, ubiquitination, acetylation, and we are more interested in the glycosylation, uh, how this receptor get glycosylated, in particularly fucosylation, which is also like type of glycosylation, and which happen with the presence of fucose, which is produced by microbiota in the intestine, and in presence of fucosyl transferase enzyme is like different kinds of enzyme. You can see over here FUT8, FUT2, FUT1, and then this enzyme convert this fucose added to glycoprotein, glycolipids, or free oligosaccharides, and this like the fucosylation happen in the receptors. Conventionally, the fucosylation of the receptor in the homeostatic is like beneficial, is like required for the immune system, and in particular you can see it actually there is some science paper that shows like a fucosylation of the epithelial cells, and actually inhibit the pathogenic or salmonella or invading like pathogens. Although the chronic fucosylation, like the constantly fucosylation, is pathological, and not like many studies have shown like how this chronic fucosylation can drive to the cancer condition. And so we hypothesize whether dysregulated epithelial fucosylation promote CD8 T cell exhaustion, which drive immune system immune evasion of the tumor cells in the colon cancer. So we we test the hypothesis like in two approach and in approach one we look at like more mechanistic approach like how to use the mouse model and like look at like how the epithelial fucosylation, how we can modulate the fucosylation. And in second approach we look at like how the using the North Dakota regional like colon cancer sample and looking at more like a tumor microenvironment and also like using the machine learning approach like to predict like more biomarkers. So recently we published one study in JCI Insight, and what we found, like you can see over here, like this fucosylation we measure with the one dye called like UEA1, like, and then we stain the different stages of colon cancer. As you can see, like the fucosylation is increases like with the stages of the colon cancer, and once we inhibit this fucosylation with a drug called 2FF, and which leads to like decrease the fucosylation or like sorry like the decrease the cell proliferation, as you can see over here, and also like some fucosylation gene as well like a cancer gene, like so just suggesting like this fucosylation is causative for the tumor cell growth. But as you can see like over here, this is like very crude way like how the epithelial cells are like showing like all entirely epithelial cells and which epithelial cells are involved in to find the more like more deep inside. So we look at like single cell RNA-seq, and what we did like over here, this is from the intestine from the young versus old and like control versus tumor. I show like representative data over here. What we found like when we define with the different cellular subset, so fucosylation is more like predominantly is like on epithelial cells. As you can see like the different fucosylation fucosyl transferase enzymes over rich all over here. More interestingly, what we found that in the when we look at like normal versus tumor condition, you can see like FUT8, which is like already people have shown like that this is involved in the tumor condition like or cause like colon cancer, and which is like more in the old condition as compared to this young. And also like FUT2 is also increased like in more in the old. So, and interestingly, if you look at like then we further like make like this subcellular the divide of the this cluster for this epithelial cells, and you can see like the how this epithelial subset is like differentially fucosylated. And in particular, for example, like is for example like CE cluster epithelial 1 or 2 is like highly fucosylated, and maybe like the more like the TGF beta signaling or TNF signaling. So we can really like it's like hard to deplete entire epithelial cells, like what you can with the gaining like more deep knowledge like how to epithelial cellular subset we can deplete, which is like maybe pathological, which could be like the interest for more like looking like more investigation. But like what I show here, like what I found like here, interestingly, like the IL-22 receptor signaling is like increased in the, this is like the old versus young in the normal conditions. In the aging condition, IL-22 receptor signaling is increased. So we validate this like why this and IL-22 receptor signaling, because there is some reports that shows like IL-22 receptor signaling in actually regulate the FUT2 and FUT8 expression level. So we look at like a mouse model conventionally where we inject like AOM, which is like a cancerous substance, and like we make like 3 rounds of DSS, which cause like the colon cancer condition. And as you can see, like young versus old, like the old has like more fucosylation, and which is pretty comparable with the human. Like if you see like the old colon cancer condition has like more fucosylation as compared to young. What is more interesting to us, like the IL-22 receptor signaling is more in the tumor condition, as well as the FUT2 and FUT8 in the tumor condition. So we look at it further, and what we found like in the particularly the PDL1, which is a, so this is like a sorted epithelial cell, EpCAM positive epithelial cells. What we found like the PDL1, which is the ligand found on the epithelial cells, and which bind with the PD-1 which found on CD8 T cells. So this is like the suppress the CD8 T cell function, as you can see over here, and this effector T cell are get exhausted. Like so, this effector T cell like make like no more exhausted. I mean like no more effector, so they do not kill the tumor. And so fucosylation, what we shows like here, like with the IL-22 increasing dose, it increase the PDL1 expression as well, and inhibiting IL-22 is decrease the PDL1 expression. So this actually like support our hypothesis how IL-22 is like regulating PDL1 expression in the once it's like the increase in the aging condition and old condition, and is regulating the PDL1 expression, and how this affecting the CD8 T cell. So I do not show here like the more functional assay. I need to like the more validate like the CD8 T cell function, like how this perforin enzyme and like more phenotyping. But up to now we are like doing that, like, but we conclude up to here like the how the epithelial fucosylation is related to CD8 T cell exhaustion. And coming on the next approach, next aim, like so what we did, like our approach, we collaborate with Dr. Stika at Sanford Clinic, Fargo, and what we did, like we make like 20 young, 20 old patient, and adjacent versus tumor biopsy, and what we and also like the PBMC also like that to look at like CD8 T cell like function are also, and we propose like a like special transcriptome over there. So this is my strategy, like the so we adopt like a special transcriptome as like cos mx. So, like those, so this is like the technique where you can really especially like, so idea is like we want to see like a epithelial and epithelial immune axis, like how epithelial cells or subset are like related with the CD8 T cells. So my hypothesis is like the fucosylated versus non-fucosylated. If fucosylation is actually causing the promoting the tumor, so in the tumor, fucosylated region maybe have more exhausted CD8 T cells. So I want to see like that, especially because like most of the technique, once you make like qPCR or like the some single cell, you make like the single cell suspension where you lose the location of the immune cell epithelial cells. So this technology is like other technology called like the Visium, which make like or like Geomax, which make like the circle or ROI. This give like information of the ROI, but this give you like actual cell information where you can see. And also like the same slide, what I'm going to do, like I'm going to take this patient sample and then slice them for same section for H&E and immunofluorescence as well as Cosmex. Then I can merge the all the sample and like with the machine learning, or I can see like the the how this fucosylated region or like whatever the cells, so which is like the avoid the like the people always like say oh is like noise or something like so I can like really with the immunohistochemistry I can say like validate my data. So here my approach is look at the aging uh, young versus old different group, and like or tumor versus match control. So I will look at it like the cell states, spatial niche, biomarker, therapeutic targets in this condition. So what we did actually, we make like a established like one pipeline like the how to. So this is showing like the here is like the tumor tissue is like the is the normal, and this is a tumor, and this window is showing like over here you can see like a these are the based on the this marker, as you can see IL-22 receptor low, fut low, and like these four marker, and like these are color coded, and as you can see like over here how these cells are situated in the spatial setting. And out of that, like the you can see here like how IL-22 receptor expression, fut2 expression, fut8 expression. So you can like really overlay, because this is like same section over here, and you can see like IL-22 receptor downstream signaling STRAT3 or like some cancer gene make or like BCL2. So so I'm going to like the make like this is like the I'm representing showing like so up to now I collected we proposed like a 20 sample, like we got like 10 samples. So I hope like I will get like the soon like the all the samples. So just to match the n number, put like all the slides like 20 patient like the adjacent like versus like tumor sample, and look at like how in the North Dakota colon cancer sample, and then how this is like the epithelial immune, like what is the transcriptome, like, and look at like how this is like the, I want to see like the whether IL-22 receptor high, fute high, and like whether this is co-localized with a CD8 T cell or PDL1 expression. So whether this is like the really like making my hypothesis clear. So so next part of that, like the, so that part like we are doing like, and once we get like all the sample, we will like complete that one. And so this is the next part, like how the, so cancer, we know like, this like multi-parametric, like multiple factors, like environmental factor, a lot of like different genes, and like there are a lot of data is like there, like, and you can compare like with the only univariant like the, but like machine learning is like really good technique to multivariant like a component. You can like really genomics, proteomics, or clinical data like integrate. So recently we published like one data like how it's using machine learning, how the age stratified like analysis of therapeutic or glycosylation and immune signals. So what we did, like we took like the two data set. One is like uh, GSC, like this one, just like the discovery data set, and we find like some marker and make like the validation. So here is a this is like the discovery data set. We find like the different genes, and in this data set, like we actually like make the validation, which make like very nice. And out of that, like we found like the different pathways, like as you can see like over here. So we targeted mostly like the therapeutic, immune, and glycosylation, how they correlate each other. So in summary, like what I show, like IL-22 is critical for in the tumor, and is increase like FUT2, FUT8, and PDL1 expression, and blocking IL-22 receptor can like inhibit the fucosylation as well as inhibit the PDL1 expression. And machine learning is like really robust technique for predicting multivariant parameter. So here is my future direction. Like I'm based on that, like I'm making like a how IL-22 receptor signaling influencing epithelial cell fate. Like so, we are looking like based on the this primary data, like how the epithelial subset and like immune epithelial interaction, and then we look at like how this fucosylation of the receptor, how stabilizing the signaling, like IL-22 receptor signaling in the tumor versus non and the young versus old. And then can we use like IL-22 receptor or like fucosylation like the therapy resistant. So we actually make like the collaboration with the UND doctor in the proteomics with Dr. Golokov, and we are plan to IP the IL-22 receptor and like do like the bind the mass spec and find the tumor specific binding protein, and like how this is like resistance, how they select the new partners. So idea is like we look at like a how the IL-22 receptor, like the glycosylation is like making like FUT2 or FUT8 expression. And we have like some other data like show like how this is inhibit the TNF like TNF signaling and make like these cells like more cell death resistance. And we also collaborate with Dr. Khushro at the University of South Dakota, and he helped us to make the organoid system. So we are going to like the CRISPR knockout FUT2 IL-22 receptor 8 to in order to like the more functionally whatever like the, I show like the special transcriptome. So the same patient data, like the we can put like the more like organoid and like the look at like the make the more biochemistry and like the functional assay in this condition. And I would like to thank like really Transcend like this giving me this grant, like the I'm really like fortunate, like, and then we generate a lot of data and like submit R01 or 21 for this like published paper. And more importantly, like make like a lot of collaboration, Dr. Stika and like Dr. and University of South Dakota also, like, so that's like really great. So and this he's my mentor, and I would like really appreciate like the research design and compliance team and administrative support, Savannah and Miranda. Thank you very much. I will have to take on.
Now, can you tell me something about the protocol that you're using to get samples? Are these samples from the same place in the colon?
So they are like getting like I don't know like the which place they are giving. So they are from the colon. We ask like the, so our like the requirement was when we ask like the, so stage 2 to 4, and this is like the like active, like the, so we don't know like the which part of the colon, but like this, we are like, our question is like looking like tumor microenvironment, like epithelial immune axis, like wherever like that, this like so.
Yeah, because I don't know that much, I don't remember that much about colon cancer, but the left and right colon are really different. And I'm just wondering whether or not it's something that you want to balance out and make
Yeah.
sure that you're comparing.
Yeah, I can, yeah, definitely. That's a good point. I think I can talk to them like about that. All right. So thank you so much.
You.